Welcome to SciGest, your fortnightly science from the Bioeconomy Science Institute.
Kia ora koutou, welcome to an episode of SciGest, digestible science from the Bioeconomy Science Institute. My name's Rebecca Bloomer. I'm joining you from our Lincoln campus, and today I am super excited to have two guests from our Manaaki Whenua Land Care Research Group. We have Dr. Jessie Prebble, who is the curator of the Allan Herbarium, and Ines Schoenberger, who is the collections manager for the Allan Herbarium. Some of you may have caught earlier this year a podcast by my colleague Hilary Ireland and Dr. Bevan Weir on the collections of national significance that the Bioeconomy Science Institute hold. I really enjoyed that podcast, but I am a massive plant nerd, and I thought it would be really nice to take a little bit of a deeper dive into the Allan Herbarium here at Lincoln. So really excited with the podcast today. Welcome, both of you.
Thank you very much for having us. It's really exciting to have the opportunity to say something about our marvelous Allan Herbarium today.
I'm so pleased that you feel that way, because I was lucky enough to have a tour. Actually, I've had two separate tours with both of these folks in the last 12 months or so through the herbarium, and I think you've both got some amazing stories in science to tell us about. I wondered if, for both my sake and the sake of our listeners, we could maybe start with each of you briefly introducing yourselves, and particularly because I don't really understand this, tell me a little bit about the difference between your two roles within the herbarium. So maybe, Jessie, would you like to go first?
Sure. Kia ora koutou. My name's Jessie Prebble. I'm the curator at the Allan Herbarium. That means I'm responsible for the scientific side of the herbarium. So when people think of a herbaria, they're often thinking of the dried pressed plants and the collection itself, which is obviously a super important part, but an equally important part is what we do with those records and the cool science that we can do and how we link those two things together. Also part of my role is outreach and staying connected with the international collection of herbaria, because of course we are not the only herbarium. There's a whole network both within Aotearoa and internationally, and we work together really closely.
Sounds good. And Ines, how about your slightly subtly different role as the collections manager? How long have you been doing that for?
I have been working at the Allan Herbarium since 2004, when I started as a collection administrator, and I became the collection manager in 2008. And my main purpose is looking after the day-to-day activities in the herbarium. So from accessioning specimens, from turning plant material into permanent vouchers, to coordinating volunteers who are helping us with this task, as well as overseeing our loan procedures. So we are connected, as Jessie mentioned before, to all other herbaria in the world. And we are like a big library. We are interloan specimens from time to time. So I'm overseeing this process to send specimens and to receive specimens from other countries. There is so much to do every day that it's really hard to outline, because getting a collection of 800,000 objects in order takes a lot of work. And we do a lot of work with our volunteers, but we also have some technical help or curatorial assistants to help us with this. And I'm supervising the technical assistants as well as the volunteers in the herbarium.
That sounds like a huge task, and there is quite a lot to unpack in that relatively brief statement. There's some words in there that actually aren't super familiar even to me as a plant kind of adjacent person. So taking a quick step back, what actually is a herbarium? Maybe a big step back. Sorry.
I always when I try to explain what a herbarium is, I remember what someone told me many years ago, and they said, "Oh, you're working in a plant morgue." And this makes me a little bit upset, because a herbarium is so much more than it's just a storage facility full of dead plants. Yes, it's correct that our plants are all dead, pressed and preserved, but they are also mounted on acid-free archival material. They are preserved as permanent vouchers to document the flora of a country. They are also aimed for identification reasons. We keep them because we can identify other plant material based on the accession plant material. We have all sorts of application for our plants. So they are not just dead plants mounted and pressed on a sheet. There's so much more. And I can be here for hours to explain. There have been actually publication done on the usage of herbaria. And one of my favorite from many years ago was 101 usage of a herbarium, which I can recommend to read. Because we have, of course, Jessie would bring in some more scientific aspects, but we have people coming, artists coming to look at our specimens to be inspired. We accession plant material from pharmaceutical companies who used our plants as for their potions and lotions, and they wanted to voucher their research. We have biosecurity officers giving us plants they find in New Zealand, which no one ever has seen before. Is it a nasty weed coming newly into a country? So we can, I always say, without bragging about our collection, we can almost answer any question to do with plants based on our collection.
It's really, I mean, maybe this oversimplifies, but it sounds like something much closer to a library than a morgue.
It is, definitely. And people not only say it's a plant morgue, they also say it's a final resting place, which gets me really upset because it's never final. Our plants as research objects, they are not artifacts. They are not things which sit in dusty old cabinets and someone comes and looks at them from time to time. They're actually actively used. They're sitting there maybe on a shelf, but someone comes along to study the plant. Someone comes along and extracts pollen from them to compare pollen profile from a swamp. Someone or research pollen for analyzing pollen they found in fossilized coprolites. They can use our plants for extracting DNA. This is what Jessie does on a regular basis. We can answer questions, when did a plant come to New Zealand? I'm talking here about exotic plants. When did certain weeds come into New Zealand? We can even answer a question, how did they come to New Zealand? Were they introduced in baleage in old ships? Did they come through harbors? Did the spread of these plants come through ports? Were they smuggled into the country? Did they come as seed contaminants? All these questions can be answered from our herbarium.
And thinking back to the tour that you gave me, one of the things that was quite striking was you have, I think, a map on the wall showing where all of the other herbaria are. I would hazard a guess that you have specimens that are better travelled than I am.
Definitely. Some of our specimens go all over the world. We are organized, as Jessie mentioned before, in the New Zealand Herbarium Network. A little bit further, we are organized in the Australasian Herbarium Network, and we are organized in the International Herbarium Network, which holds about more than 3,500 herbaria now. And in theory, we are interacting with every single herbarium of these 3,500 registered herbaria. We are allowed to send specimens to other herbaria for looking. They're usually not allowed to touch, extract DNA or do anything with them, but they can use it as a reference material and can look and compare with their material. In some circumstances, we can give them permission to extract DNA or do further research, but this is, we need permission for this. So we are engaging at the moment with about 1,000 different institutions as today, that they have specimens from us or we are getting specimens from them in. So this is another application many in the BSI don't know. If you want to have a plant for research, if you need a specimen to look at, to compare, we can get it in for you, as long as it's dead and dried. We're not importing fresh material, but our facility allows us, as a containment facility, to get material in from all over the world for you to compare our specimens with, for example.
So to stretch my kind of hacky library metaphor, something akin to an interlibrary loan.
Yes.
Is it a terrible metaphor?
No. It's perfect.
Yeah. Okay, good.
Except they'd get unhappy if you rip bits off library books.
Very cool.
Even with permission.
Very cool. Jessie, the international kind of connection side of it, a lot of that, I guess, would sit within your outreach role. Is there anything you want to add to that?
Yeah, there's various parts of it. So the connections with Australia is probably the relationship we put most effort into, because that's where we have a lot of the similar issues and, similar opportunities, so we work together. But internationally, there's every 6 years the large botanical congress and the nomenclature session, which is another really important part of our field to be involved with. That's where we can update the code of botanical nomenclature. So one of the things that we're often involved with as taxonomists and systematists is the naming of plants, which is a really often fun part of the job. And those international connections and collaborations help strengthen the rules around how we name those plants, and we work together.
I imagine that can be a reasonably vigorous discussion process at times.
Yes, and extremely nerdy.
Yeah, most of the best things are. Enos, this reminds me of a story you told me about renaming of a particularly large group in New Zealand, and how you wanted to wait a little while to make sure that everyone was definitely happy with that name before completely arranging the collection based on its alphabetical order.
That's correct, yes. So we always endeavor to have the plants named and labeled to the latest nomenclatural rules, to the latest name which is preferred in science. And one example is, for example, when the name Hebe changed to Veronica. It's a very large genus in New Zealand with many, and I don't have the exact number, species. And we have about 2 rows of tall herbarium cabinets full of Veronica specimens, Urbex and Hebe specimens. So now we had to rename every single specimen into Veronica and shift entire rows of herbarium cabinet content from one end to the other, because this is how we file things in alphabetical order in the herbarium. This is very tedious. And lo and behold, I came across some specimens which were clearly Hebe, as I would have known them, but they were still labeled Veronica. Because originally...
from the last time around.
That's correct. Because before the genus Hebe was erected in New Zealand to cover the New Zealand unique-looking plants, specimens were known, or species were known as Veronica, as, like the ones you find in Europe. And so I still had specimens which have never been touched, which has never been named Hebe, still sitting under Veronica. And so they missed the big Hebe name update, and now I didn't have to update them back to Veronica. So sometimes I say, if you wait about 150 years before you spring into action, things sort out itself. So, but I must say we did the change because it's to change all of our Hebes into Veronica, but this is based on thorough research and scientific information. So this had to be done in my eyes.
Kind of raises another interesting point that I don't know if everyone would be aware of. I had thought of a herbarium as full of, I think it's what you call vouchers, like the literal, a dried pressed plant on a sheet of beautiful archival paper. But that's, of your 800,000 objects, that's quite a lot of the collection, but it's far from all of it, right? There are lots of other things that you hold and other ways you preserve things.
That's correct. And I sometimes tend to confuse people because I use different terminology. I call them specimens, vouchers, plant material, or objects. When I talk about 800,000 objects, the majority might be pressed and dried plants on an archival sheet, but about 1/3 of the collection are not on big sheets, but in small envelopes. These are our cryptogams. Imagine you have a little lichen, moss or liverwort, which is just a very tiny sample. It would not make sense to mount it on a big A3 sheet, but they are stored in archival envelopes. So lichens, mosses, liverworts are the really important plant groups in New Zealand. So we have more than 800 New Zealand native lichens. We have more than 600 liverworts indigenous to New Zealand. So people overlook sometimes. I think a herbarium is just about grasses and trees, but these are very important organisms for New Zealand.
And there are things that you make a point of storing in ways that keep their three-dimensional structure too, right?
Correct.
I was kind of blown away when I walked past a room labelled the spirit room. Like, what are you guys keeping in there? But that's where you're storing flowers and things,
yes.
right?
most of the time, and let's say 90% or an even higher percentage, plants, all the important characters are preserved by simple drying. And we'd like to do this because DNA is still available. We can see still structures and microstructures in a normally pressed and dried plant material. But sometimes it makes no sense. Let's say if I have a beautiful lady slipper orchid and I squash it in between the newspaper in a book or in a herbarium press, then I have a big blotch on a sheet of paper and I can't see the three-dimensional structure. And especially for orchids, it's so essential to have some of these three-dimensional structures preserved for identification reasons. In this case, we like to pickle it. So the spirit collection is sometimes called pickle collection, wet collection, or my preferred name is spirit room because it has sort of the spiritual aspect to it. And so we put it in ethanol. It's usually 70% ethanol with a little bit of glycerol to keep the plant material still flexible. Unfortunately, all the color is washed out. They look a bit ghostly. But we usually don't just keep it a spirit sample, we link it to a pressed sample. So it's all connected, interconnected. We also like to store fruit or some special berries, for example, in spirit collection. We also have cacti in spirit collection where we preserve a three-dimensional structure of this. They look a bit ghostly and white and washed out, but imagine if you press a tomato, which is the fruit of the tomato plant, on a sheet of paper, then you have tomato sauce on a sheet, which makes no sense.
Yeah.
So if you keep the whole tomato in a pickle jar, then you can examine it better. So yes, the spirit collection is one ancillary collection which is linked to the herbarium sheets, usually to some sheets containing leaves. But it's only one of many ancillary collections we have here. We have also a wood collection. If a chunk of wood is really sometimes hard to press or to put on a sheet, so we have a wood sample collection. We have a microscopy slide collection associated with wood samples. So sometimes 3, 4, 5 components are all merged together from one plant in different storage areas of the collection. Obviously, the spirit collection can't be held right next to the dried plant collection because of fire danger.
Yep, I hadn't thought of that angle, but that is reasonably important. There are some phenomenally cool specimens in there. Do each of you have do you have a favourite?
I mean, it's so hard to choose a favourite. I'd say for me, my favourite, it changes all the time, essentially, depending on what I'm working on or ones that I've recently found or cool stories that I've come across. And at the moment, one that would maybe be considered a favourite is, you would never think it was a favourite to look at it. It's completely unassuming. It's just an indigenous shrub of Pimelia, which is sometimes called New Zealand Daphne, because it's in the same family. It doesn't smell like Daphne, unfortunately. The flowers are not pink, they're just small and white. But, beautiful shrub found throughout New Zealand, lots of different species. And we got asked by the Department of Conservation to look into this particular one. It's a critically endangered plant. They've been looking after it as one of their species on the brink, which means it's not only critically endangered, but they're actually doing some active management,
right,
looking after the species. They'd been looking after it in this way since about 2011 when the species was described. But they were starting to kind of have some doubts about the whole process. There were only about 7 plants left in the wild. They've been growing them on, but the plants they flower pretty well, but they only fruit really occasionally. And then those fruits don't usually actually develop into seedlings, or when they did, they all died out. So they were kind of saying, is it so inbred, perhaps, that it can no longer set seed? Or is it a hybrid? Should it never really have been considered a species in the first place? And we'd been doing some work on Pimelia anyway, so we'd developed some genetic markers. So we were like, well, we can't promise anything, but we can test it, and, if you can get us samples of who you think might be the parent plants and any other plants growing around, we can do some genetic work and have a look. And it was the clearest structure plot I've ever seen, 100% hands down a hybrid. And when we went back through the herbaria to see, what we had in the collection already of this plant, there was a specimen from 1979, collected by Tony Druce, who's a big name in botany in New Zealand. He'd collected a plant from this location, and the name he'd given was a possible hybrid of,
So the clue
of
was
these
there.
two particular species. And so it's so cool. We so often come across these things. We'll do something and we'll do the research, and we need to do the research anyway, but actually you go back to the herbarium, you find these specimens. Yeah, the clues are there. The past is talking to us.
Yeah, it's really, really nice, I think, it's not often that you get something that shakes out so incredibly cleanly too, but to then have that validated with, actually, it turns out someone had thought this looking at it in its original collection site too. So your favourite specimen is that original collection?
Yes.
That's very cool. I like that. What about you, Ines? Do you have a favourite?
God, it changes on a daily basis.
This morning when you came in.
Well, I, of course, appreciate the specimens, the older specimens we hold at this collection. This is 91 sheets or specimens collected on Captain Cook's first journey by Joseph Banks and Daniel Solander. Sometimes I'm very proud that we have this here at Manaki Whenua in the Allan Herbarium. They don't look spectacular. They look exactly like the little sage you collect or the manuka you collect these days. But I know that they are more than 250 years old. That makes it so special for me. I know that they have been on the Endeavour. They have been dried and touched by Joseph Banks and probably Daniel Solander, maybe by Captain Cook, we don't know. But they make it really important collection for us because they indicate what was here before Europeans came. So all of these collections are indicating New Zealand, definitely New Zealand indigenous plants. And they look in pretty good nick. So it just show you how well, if you look after the plant collection in a dry conditions, climatized rooms, archival materials, they can last forever without any degeneration. So they're really good.
It's quite incredible, isn't it, that they can last so well. And those ones in particular, I can't imagine had an easy start to life. Imagine trying to press and dry plants on a ship.
I think that some of those ships often turned up with just holds of rotting vouchers.
Yeah.
So, yeah.
Yeah, I can imagine. Because what does the process, in an ideal world, which hopefully we can at least get somewhat close to here, what does it look like to preserve a specimen on site here? Clearly not like a ship's hold.
It would be very similar. So we actually use the same material. We use newsprint. We try to tend here unprinted newsprint so that the print doesn't transfer to the plant specimen. We put it between wooden boards and some spices, cardboard in between, and put it into a herbarium dryer, which is a little, it's quite fancy, because if you want to preserve a plant, it's nothing wrong by just drying it gently on the bench, and just make sure that you change the moist paper in between and keep it dry. And this is exactly how they would have dried it in the Endeavour, except that the Endeavour was a bit wetter, I guess, than our herbarium dryer. So we never dry it or crank, don't put it in a baking oven. So I've come across a lot of students while working here, and they had to do some collection for their assignments. And they're never on time, and the plant takes a few days to dry, and then they pop it into a baking oven and turn it up and dry it at 80 degrees, and the plant comes out really crinkly and crumbly. So, and the DNA is wasted, of course, and some of the characters might be gone. So the key is dry it gently, carefully. Change the blotting papers around it, but just don't dry it in a higher temperatures than 30-35 degrees. Otherwise, it gets all crinkly and you have dust on a sheet of paper. So
I think Ines may have some insight into my 200 level flora of New Zealand assignment that I didn't perhaps need shared with the world. Thank you.
We all did this. So I can talk us out of experience. When I had to do my first herbarium, I had put it into plastic sleeves, and they weren't just quite dry, and so there was a lot of mold left.
I suppose at that point you're kind of thinking, well, I just need to pass my assignment. But when you think about it as something that might 250 years later be useful to a botanist in a later life.
And some
not
students
just a botanist.
Obviously as well have managed this, because we actually have quite a few collections from students, even when we've kind of assimilated some of like the Armstrong collection and various collections from around the place that have student collections?
This is it, right, because I always say, being a collection manager, you have to be a hoarder.
A hoarder.
Yes.
I think that's true of most kinds of being a scientist, actually.
Every single specimen is an object. Every single specimen tells a story. And there's nothing, there's nothing the same. Yes, I collected a grass here on the Lincoln campus last year. Is it still here this year? I want to have all different scales. I want to have one collected by Banks and Solander and one by you tomorrow. I want to have one collected in spring, one in autumn, one in summer. I wanted one from Cape Ringe, one from Bluff.
They're all telling really different parts of the story,
exactly.
I guess, aren't they?
So I would like to have because a lot of people come and say, why do you keep all this stuff? And I say it because they're all unique specimens. They all tell you a story. They all show different characters. I want a kowhai tree with big leaves, one with little leaves. I want one with flowers, one with seed pods. So it's never ending. And a lot of these plants, and when people come, like students or smaller herbaria from Department of Conservation, some student likes the Mount Cook collection or the Arthur's Pass collection, and says, do you want this? And I always say yes, because they bear
we'll find a home.
Yes, and they bear some information or some distribution gaps we didn't have.
Yeah.
A very key point of a herbarium is that it documents the flora of a country, and we want to have one from everywhere where it grows, even remote places.
I think something that's really hard to know too, and maybe this is a good segue into some of the science stuff, is it can be so hard to know how something might be useful later. I had a really interesting chat with another Manaaki Whenua person earlier this year for another podcast, Pierre Odier, about the soils collection. Cool. And he was talking about how that had been kind of rescued, I think, around the time the DSIR was disestablished and we became the Crown Research Institutes, and there had been these jars of soil collected since like the early, I can't remember, sorry Pierre, but it was quite a precious collection, and someone had the foresight to go, no, we can't chuck that out, and it's actually been incredibly useful since.
Well, there's been huge changes in how we use the collection. 60 years ago, would anybody have thought that we would be able to extract DNA from a herbarium specimen and in a useful way and do cool things with that? Probably not. And also with all of the digitizing that we're doing, and databasing. So our collection is not yet fully databased. That's still a work in project. And as we're doing that, as we're databasing them, we're imaging them, and then we're sharing that data. And we share that in quite a few different ways. We've got our website, so you can go and, if there's a particular plant that you're interested in, you can just search for its name and you can get a list of all the specimens we hold, and you can look at that in a map view and you can see how they're distributed. But then, of course, that's just our collections. If you want to see that aggregated with other collections, you can go to the Australian Virtual Herbaria, or you can go to GBIF, and all of our data goes to all of these kind of aggregated places, and then people use that data in ways that we never would have thought of. Like heaps of really big ecological, worldwide studies use our data, and you can't predict which specimens are going to get used, which things people are going to find useful in the future, other than, as Ina said, trying to get a good a coverage as possible through space and time. Yeah,
and season and all the different kind of aspects that there are to that. Using that as a sort of chance to segue into some of the science, have you got any favourite projects at the moment, Ines?
I love it can answer so many questions, and while I'm not doing the research myself, I supply material for research. So if a biosecurity officer comes and says, has this plant ever been collected in the South Island, I can just show him in our collections where we have found it previously. If someone comes in and brings a plant which has never been collected in New Zealand, that's also very exciting.
Exciting and sometimes a little bit too exciting maybe.
And I just recall when the minister came, looked at the collection, and he asked me a question. I said, how many new plants do you discover? And I said, well, we still describe a couple or 3 or 4 species a year from New Zealand native plants, but we are discovering many, many more exotic plants which never have been recorded in New Zealand. And most of them are not super exciting because they are not agricultural or economic important weeds, but they can be weeds for environmental weeds problems. And we have first records of many, many plants in New Zealand. And not that every week, but every month we get a plant which has never been recorded in New Zealand before. And so it's important to voucher it, to put it in the collection for later records. Sometimes you have a plant which sits in New Zealand forever. It's just either a garden plant or it just pops up here and there, and it's a leg fast of a weed. And then suddenly it takes off. And you see this when you go around, and when you walk around and say, oh, I've never seen this before, and now it's everywhere. So the collection is used quite frequently from biosecurity officers.
You can imagine too, in a kind of rapidly changing climate situation where you've got places that are suddenly maybe warm enough for something to survive a winter.
Exactly. We can now see, especially from the weed perspective, that a lot of weeds, only known from the warm North Island regions, from Auckland, are slowly making their way down. First they pop up in Nelson, and suddenly we have some even in Christchurch, and we can monitor this spread. So we can actually map this distribution, and this distribution changes.
And potentially, I guess, if people are looking in the right place at the right time, that gives clues as well to where you might see it next, and perhaps a bit of a heads up about where it might become a problem.
Exactly. And then the biosecurity officers, especially from the regional council, put things onto their watch lists. And there are actually some of people supplying specimens for us, because it's really important to monitor. I'm not out there, so I don't leave the herbarium much. I'm looking after the plants when they're submitted. But I depend on people with a keen eye on plants to submit these specimens to us so that we can enhance the collection.
I don't know which of you this is a question for, but how do you see the herbarium as being influenced by collections alongside things like iNaturalist? Like, if people are putting something into iNaturalist, I guess there's a subset of those folks who would still feel super motivated to put it into the herbarium as well. They're adjacent resources?
They're adjacent, yeah. They work together. We actually really appreciate iNaturalist and use it a lot if we're planning field trips, for instance, because people are more likely to take a photograph of something and put it on iNat than, as you say, go through the process of getting a permit perhaps to collect and then to submit, because of course depending on where you're collecting you do need to get appropriate permissions.
Yeah, of course.
And if we've got both, if we have an iNat record and a voucher, we can link those up in our database and have them talking to each other. But yeah, the difference really is that having the vouchered specimen means that in the future, if that name gets changed, if someone comes along and looks and has done some work and realised, actually, no, we thought it was this species, but now it's this species, we can make that update in our database for our record. iNat doesn't, you don't have the physical specimen to interrogate in the same way.
Yeah, sure, and it's not a resource that's available for DNA extraction.
It's not, and you can't look at a lot of characters in a photo. A lot of characters you can see from a photograph, a lot of characters you can't.
Yeah, sure, which, I mean, talking about characters maybe is another useful segue. I am the world's most rubbish botanist. You are not. So can you tell me a little bit about the importance of the herbarium for systematics work?
Yeah, I mean, it's super important. It's the basis of a lot of systematics work. So morphology, what the plant looks like, is still, even in this day of DNA, is still still the primary character that we use when we're looking at species and at plants. And if you're undertaking a research project, you're literally going to be measuring characters from herbarium specimens. You do that alongside looking at live material and hopefully growing some if you can. But we'll literally sort out as many specimens as you can of each species that you're interested in and figure out which characters you want to measure and have a giant spreadsheet and be measuring sometimes hundreds of characters per individual in order to build up your data set, your morphometric data set that you can analyse and then try to figure out where you think the species boundaries might be.
And I guess if you find something that looks a little bit different on you, plants are pretty weird. It can be hard to know sometimes, is this just normal variation within something we already know, or is it genuinely novel?
Absolutely.
And having that resource, I guess, gives you a lot broader set of comparators you can look at. Hopefully.
Exactly. Yeah. So you want to be able to compare what you've found to other things similar to it, and you want to be able to compare as many different character sets as you can. So the morphology combined with the genetics, combined with chromosome counts if you can, combined with growing live plants, gives you a lot more power to help to figure out those kind of questions.
This is an incredibly silly question, considering it's not unadjacent to my own kind of research, I guess, but you can certainly get usually DNA out of even pretty old herbarium specimens. Can you get chromosome counts? No.
Okay. No, you wouldn't need live plants for chromosome counts. And actually, yeah, the DNA you can get out of old specimens does vary widely in how good a quality it is, how useful it is, what you can actually do with it. Yeah. And then once you've done that kind of work, you've probably published a taxonomic revision, and then you're going to be starting to think about a flora.
Yeah.
And writing floras has been a huge part of the history of the Allan Herbarium. The paper hard copy volumes, we've got volumes 1 through 5, and then the lichen flora.
So the lichen flora is not part of the plant flora, obviously. That's a separate.
It's a separate one.
Yeah. But it's in
the flora series.
It's in the flora series, yeah. And we're currently working on the liverwort flora series.
That's cool.
It's very cool. And we've also kind of moved on somewhat from these, working on the flora of a whole area, writing a physical book and publishing it once every 50 years to trying to do this in a way where we can update it slightly more frequently and easily. So we've got the e-flora, the online flora, which we can publish things by family. And so you can find all those resources online.
Well, we can link to all those resources via our podcast page too, if that's useful, including actually note to self. We should make sure that we put the 101 uses for herbaria on that as well.
Yes.
That'd be quite fun.
Yes. Herbarium always struggle with funding because they can be a bit expensive to run. You have a special customized room, you have air conditioning, you need high quality archival material only, you need qualified staff. And this can be, people say, it's a lot of money. And this happens to all collections worldwide, so it's not just our collection. But there's a lot of interesting material out there, publications very recently about the change of herbaria, the usage of herbaria, what incredible resource a herbarium is. So other things are, you can sample herbarium specimens for plant pests. You can sample for viruses. You can look when was this virus, and it's not just the main sheet, of this particular species. It is maybe it has a rust fungus on it. So we do not sanitize our specimens. They come in, they're pressed, they're preserved as they are. And you wouldn't believe, but one day a person from a research institute came to look at Antarctic mosses and lichens and liverworts for tardigrades hiding in soil. You wouldn't believe how many people come and ask for weird and wonderful things we can help them with. As I see, an artist coming to be inspired, someone just looking for insect frass. I have plants chewed on by kakapos from Fjordland, kakapo browsing from the wild on Asifylla. It's just amazing when you go through the collection, and find these things where there's an this one was collected in the 50s in Fjordland, and it has clear kakapo browsing on it. So it makes wow, this is a real connection.
So it can tell us maybe a lot about species distributions that we didn't mean to collect for too, I guess.
Absolutely.
Just
really
accidental.
cool. Yeah. One thing I guess that's front of mind at the moment when I get now to do these podcasts with some of what were previously separate CRIs that we're now merged together with is we've got this kind of massively increased diversity of research interests and expertise. And I'm curious whether you two have found through the merger that there are different people, or do you see different opportunities for connecting out with some of the wider BSI than maybe you would have thought of previously?
We have been previously working with some groups from the BSI, like Egg Research, big component of in identifying and describing agricultural weeds, and we have always working together. And so I think with the BSI now coming together, we have even bigger and greater chances. There's some barriers are now cut away, and we can really freely work together. And so I'm very excited about this, that people get to know us, they get to know what we can offer other research groups in the BSI, but also use us as a facility to voucher their research. It's so important. People think, oh yeah, I extracted DNA, was this component out of this plant, and then are you sure it was this plant? Mm-hmm. So if you put a voucher in our collection, you have your research backed up. People can question. So it's very important also for DNA work when you say, I extracted this DNA out of such and such plant and uploaded on GeneBank under such and such name, and some later on I come along and say, you got this wrong. It's actually not Manuka, it's Kanuka. Things like this can happen. So
well, even with putting my selfish kind of plant breeding genetics research hat on, we have things like the nationally significant germplasm collections.
Yes.
And you'd think that there's potentially a really nice kind of natural interface there where, you know, the intent is obviously always to keep those as a living resource. That's kind of the point of them. But also having that backed up in the herbarium as well might be something. I don't think that's something that already happens, but maybe that's the kind of thing that could be a useful link to make.
We have already, we are vouchering, for example, the seed bank plants
cool.
so from the National Seed Bank. Of course, since it's based in Palmerston North at Massey University, they have a voucher of all of their seed plants at the Massey University Herbarium.
I didn't know that. This is the Margot Ford
Is it the Margot
collection?
Ford
Yep.
indigenous
Yes.
plant collection?
And so, yes, that's correct. And they also, for safekeeping, make a duplicate to send to us.
Oh, cool.
So we have a duplicate specimen of all the seeds held at the seed bank.
I didn't know that.
So
that's neat.
it's because it is really important to have it backed up. I can say I extracted the seed from such and such plant, but you know, people can go wrong. Things can be messed up. Names change, taxonomy changes. It might have been a variety or subspecies, and so having a voucher launched in one of the herbaria is important. It doesn't have to be with us. It can be with any of the other New Zealand herbaria. As I said, there is a really good collection at Massey University, which is within our herbarium network. It's not that I need to have everything. I would love to, but
if you could see her face right now, listeners, she would definitely like everything.
But we are only one part of the New Zealand herbarium network, and there are other two large collections in New Zealand. The second largest at Auckland Museum, and the third largest in Te Papa. We are by far the largest collection. But size doesn't matter. We always work together very well, and we're not competing. And you mentioning iNaturalist before, I'm linked to some of the collectors. There was a collector who monitored a lot of cultivated plants in reserves and gardens. He reaches out to me and says, you don't have this in your database, this specimen. And I said, oh, thank you. And I put it in our names database, but I cannot. And he said, oh, it's too expensive for me to send it down to your herbarium, but I will submit it to Te Papa. It's perfectly fine. Yeah, I can have it, and as long as I know that the species is in New Zealand to monitor it. So it's a great thing being in a herbarium. We are not alone. We are connected. We are well connected with the other collections around the country, around the Pacific area, and around the world.
I really like the sort of sense of community that comes from this, I think. Like, I don't know, it just feels nice. Between that, and I know that a lot of the work that's done vouchering things at the moment is also, that's a lot of that is volunteer led, right? So you've got kind of community within herbarium, community within the research world, this big international community of herbaria as well.
It's a lovely place.
Honestly,
Come
it sounds
visit
it.
us.
Can I?
I would really encourage, if you're interested, if this podcast tickles your fancy and you would like to have a look and might think about something we can do for you. Getting loans in from other collections, like an interloan, like a library interloan, but a plant interloan, or helping with sending some of your material away. We getting material in, for example, from other international collections from Missouri or New York for DNA extraction to compare for our wheat research. So there's so many applications for our collection. So
Can confirm also that both of these folks are very interesting to chat to and give a great tour. I have kept you both busy for quite a long time. This is getting to be a long podcast, and I should probably find a gracious way to wrap it up. Is there anything before we do that either of you feels like we should have talked about? Any particularly exciting points?
Well, there's so much to talk about in a collection. As I see it, with 800,000 specimens, there are 800,000 stories, and each specimen tells a story. Maybe the specimen itself, maybe the species itself, maybe the collector. Historical collectors we do a lot of historical background search too for great scientists and botanists in New Zealand, because we have their work in our collection, their collections. I was now where H.H. Allan, the founder of our collection, was on Christmas Day in 1928 because he collected a specimen from there, and things like this. So it's not always just about plants, it's also about people.
It's also very much the people.
Actually,
really
that
cool.
reminds me, you're saying 1928, we're going to be 100 years old, the Allan Herbarium, in 2028.
That's pretty soon. So that's really just after the establishment of the DSAR, right? Which I think is 100 this year or close to.
Yes, that sounds right. Yeah.
That's a pretty cool milestone.
Yeah.
It's going to be a good birthday party. Can I come?
Yes.
You're invited. Awesome. Thank you both so much for your time today. This has been a super interesting chat. I'm very excited to see what might come next from the herbarium, especially with opportunities in a merged BSI world. Thank you both heaps. And thank you, of course, to our listeners. You've obviously found us today, but as ever, you can find us on all normal podcast channels where you'd normally go looking for your favourite things to listen to. Thanks very much for your time. Mā te wā.